And thanks to everyone for being here. So basically this talk is going to, whoa, is this mic really loud? Is it okay?
It's good, it's good.
Okay, great. So this talk is going to be about why fish don't tend to get this big anymore. So of course that's in part a demographic effect that when we fish really hard, few individuals get old enough to get this big. But it's also increasingly being recognized that there actually might be a genetic component to this. So we all know how plant and animal breeders have been extremely successful as changing the morphology and life history of different animal and plant species to yield benefit to us. And they've done this by generation after generation, selecting the individuals with the most favorable properties and making them the parents of the next generation. It's much less appreciated that in wild capture fisheries, or even recreational fisheries, we're basically doing the opposite. So we're taking out the individuals with the most desirable traits, so the largest individuals, or the individuals that are easiest to catch, and then we leave the rest of the fish out there to pass on their genes to the next generation. So it's pretty intuitive to see that when those traits that we're targeting have a heritable basis, then removing all the favorable genotypes could cause a rapid evolution in these systems. And especially in cases for commercial fisheries, where fishing mortality is often many times higher than the natural mortality in the system. And there's more and more evidence that fishing actually has caused these kind of changes across the world's fish stocks. So here we're looking at some historical data for, some historical data from a cod population in the southern Gulf of St. Lawrence, where the left panel is showing how the length at age 6, and the right panel is showing an index of the timing of maturation. So we can see both of these have dramatically declined over recent decades. And these kind of changes have been documented for a lot of different fish stocks. And they are of concern because slower growth rates, earlier maturation might reduce overall stock productivity and resilience to overfishing. But of course, with these kind of time series of observed traits out in nature, it's really difficult to disentangle what component of this is truly a genetic and evolutionary change and what's just environmentally induced. So I was really curious to look into this question by seeing whether we can detect any signatures of fisheries-induced evolution directly at the genetic, at the DNA level. And we had a really amazing source to be able to do this, which was DNA extracted from archived otoliths. So I don't need to tell anyone here what otoliths are. These basically little data loggers from the inner ear of fish that mostly have been collected because of the growth rings that can be used for aging. But fortunately for me, and a lot of other people too that are interested in historical patterns, for many fish stocks, these otolith collections have actually been kept after they've been used for the initial aging. And here's, just to give an example, a photo from the basement at the Greenland Institute of Natural Resources, where I've been doing a lot of my work, where they have this huge collection of archived cod otoliths. Then you can see the collections go all the way back to the 30s. So this is just an incredible resource because otoliths themselves actually don't contain much, if any, DNA at all, but the little bit, basically the sloppier the technician who took out the otolith, the better, because the little bits of blood and mucus that's left there and is dry on the otolith is what we can extract the DNA from. So this means that these archives here are basically just gold mines of information about the past that we can use to reconstruct what the genetic composition of populations used to look like at certain known time points in the past. So I set out to take advantage of this by looking for genetic changes over time in this cod population from the southern Gulf of St. Lawrence that I was showing you has shown major declines in growth rate and maturation patterns. And for this particular population, we were able to find historical otoliths dating back to the 1920s, and then there was a bit of a gap, and then we had a more continuous time series from the 60s onwards. And from each of these years, we sampled around 30 individuals, so we could get pretty good estimates of the allele frequencies at any particular point in time. So we extracted the DNA and we used a genotyping chip to analyze up to 1,000 markers. So these called SNP single nucleotide polymorphisms, which basically are just places in the genome that are variable among individuals. And we then did a screen to look for which of these 1,000 markers seem to have shown more change over time than we would expect just by chance, and therefore likely have been subject to selection. And we found a total of 30 that was behaving in that way. And these were not just randomly distributed in the genome. They were primarily clustered into two big blocks. So here's just the genetic map for cod. And what's really interesting is that one of those blocks that seem to have been under selection within our century or 8-decade time series has also shown in other studies that are comparing cod populations across space, have shown that allele frequencies at this particular set of markers correlate quite tightly with the local sea surface temperature. And when we looked within our samples, our time series, we see kind of a fluctuating pattern up and down, but that actually turns out to correlate, at least for the last part of the period when we have good temperature data, it correlates quite strongly with the local temperature that these cod experienced. The other linked group of markers that we saw evidence of being under temporal selection shows more of a clinal pattern. And this particular set of markers have in other geographic studies of cod shown to correlate with local life history types. And you might recognize this sliding pattern from the earlier graph I was showing, which was showing this decline in maturation pattern that correlates really well with what we see at the genomic level. So basically, these results here are consistent with that it could be temperature having caused selection on one part of the cod genome and fishing having caused life history changes in maturation at a different part of the genome. But of course, these are just correlations and based on very few time points. So it's quite difficult to really robustly attribute this to fishing. So based on that, based on this kind of less than satisfying conclusion, I was interested in taking a step back and thinking about what kind of genomic signatures do we even expect from fisheries-induced evolution. So if we could look at this under more controlled conditions. And I was thinking back to this experiment that I remember learning about when I was an undergrad. So this was a paper that was published back in 2002. And the experiment is actually summarized well in this photo. So these fish are Atlantic silversides, and the individuals here have all been raised under the same conditions in the lab. And the differences between the three groups have evolved over just 5 generations of size-selective fishing. So basically what this experiment was all about was that they took these silversides from the wild and established 6 populations in the lab. And then they would start harvesting them. And in 2 of the populations, they would take the 90% largest individuals, kind of simulating what we might do in a real fishery. In 2 of the populations, they would take 90% smallest individuals, and then there were 2 control lines where the harvest was just random. Then they let the remaining fish reproduce to form the next generation, keeping each population at about 1,000 individuals. And you already saw how this caused really striking shifts in the growth rates of these different populations. So here you're just looking at the data where on the left side the weight of the total harvest, and on the right side the weight of individual fish, how those changed over the course of the experiment. And you can see already after 4 generations, the size of the fish that had been in the populations where the large individuals had been harvested, like in real fisheries, were only about half the size as the fish from the populations where the smaller individuals have been removed. And since these were all raised under common conditions in the lab, and the only factor that differed between them was the fishing regime, we know that these are definitely genetic shifts, and we know that they're fisheries-induced. But what we have no idea about is what were the underlying genomic shifts for this. And back when they did these experiments, it wasn't really possible to look at this in any meaningful way for a complete non-model species like these silversides. But fortunately, they had the researchers who did this experiment had the foresight to just throw all the fish in the freezer. So then now we've been able to go back and, to much dismay of various lab mates who have had to smelling these fish as I was expecting them, we've been able to recover the DNA from these individuals from the original experiment. And looking at the genomic basis is interesting just because it's just from a basic curiosity point, it's really cool to understand what's going on here, but it also has practical implications for management because looking at what genes, what genomic regions change may help us better understand the physiological mechanisms that this kind of fisheries-induced evolution can work through. And also looking at the extent to which it erodes the genetic diversity, or that we lose genetic diversity in the process, will tell us something about how reversible these changes might be once fishing stops, if we're really compromising the future evolutionary potential of the affected populations. So for this project, we took advantage of how much DNA sequencing technology has just really been revolutionized over the past decade. And basically, we were able to use this technique where we could screen the whole genome of these fish for only about $50 per sample. So we used this to analyze over 800 individuals and generated almost a terabyte of raw sequencing data that we then boiled down to look at the variation across about 2.5 million SNPs. So this is really scaling up compared to the previous study I was talking about, where it already seemed super cutting edge that we could look at 1,000 places across the genome. Now we're looking at millions, so we're getting a much more comprehensive scan. And so the first thing we did was to look at what's been changing over the course of the experiment. So we're then looking at the frequencies of these 2.5 million variants between 75 individuals sampled from the source pool used to establish the experiment, and then 50 individuals from each of the 6 populations after the 4 to 5 generations of size selection. And if we first just look across the whole genome at levels of diversity, we can see here that this is plotting the number of polymorphic sites, basically just a measure of genetic diversity. So we can see that each of the populations, so these colored bars here are in generation 5, each of the 6 populations. So we can see that there was a clear reduction in diversity compared to the initial state shown here, the gray bar, which is completely expected when we have breeding in a small population in the lab. But what's interesting is that each of the 4 size-selected lines, both the up-selected and the down-selected, show lower genetic diversity than the 2 control lines. So clearly, the size selection was accelerating the loss of genetic diversity in the populations here. So that's in part because it could in part be because the effective population size got reduced due to the selection, so there was a lot more random change. But it's also very likely that there was some targeted shifts due to selection. So to filter those 2 apart, we started looking at what changed in parallel. So which SNPs changed in the same directions in the 2 replicates of the same size selection regime, and in opposite direction in the other opposite regime. And one thing I forgot here before talking about the parallelism. In addition to the control lines showing less overall loss of diversity, they also showed much fewer SNPs with a significant allele frequency change. But I'll get back to this in a second. So these allele frequency changes, we saw that there was more parallel change. So there was a lot of random change, but there was also more parallel change among replicates than we would expect in simulations that we do, suggesting that it wasn't just a reduction in the effective population size. And so a lot of these changes, this was across thousands of SNPs that we saw these parallel changes, and none of them show that there's not like a single variant that show a super strong correlation with size. But when we look across the top percentile of SNPs that show the largest parallel shifts, we see that the allele patterns at those loci correlate very strongly with the individual size of fish. So that's kind of giving us extra evidence that these actually have something to do with the selective response. And so to further look at this, or to get a better understanding of what was going on, we also looked to wild populations of this species. So these Atlantic silversides are distributed all up and down the east coast of North America, and a lot of prior work has shown that the short growing season in the north has selected for faster growth there, while trade-offs with predator avoidance has selected for slower growth in the south. So it's well documented that there's a really strong natural climb in genetic differences in growth rates in the wild, as is shown in this pattern here. And just to give a little bit of perspective, so these are fish that we have raised in the lab, and they were spawned of wild-caught parents from Georgia and New York on the same day, and then raised under common conditions. So these are the kind of, they're close to adult size, and these are the kind of size differences that we see, even in individuals raised under common conditions. So it's quite a dramatic divergence in nature too, for the species. So to be able to compare the signatures of size selection in the experiment to the signatures of natural, so the gradient in natural selection that the species experiences in the wild, we included samples from 4 locations across the distribution range. And we then looked at the set of SNPs that both changed in parallel among replicates in the experiment and also showed allele differences among the wild population, so also appeared to be involved in the size divergence in the wild. And on this plot here, we're looking at that subset of SNPs, which is about 1,500 SNPs, how they change the direction of their change in the experiment. So I'll just show you these density plots of the allele frequency change in each population. So basically, SNPs for which the southern, naturally slow-growing allele became more common will fall out to the right side of the plot, and SNPs for which the northern, fast-growing allele became more common will be on the left side of the plot. So we can see here, the green lines here are the 2 control lines that kind of fall right in the middle. There wasn't a particular tendency to go one way or the other. But both of the down-selected lines show a very strong skew towards an increase in the naturally slow-growing allele. And both of the up-selected line show a very strong skew for the northern fast-growing allele to increase. So this is really showing us that there's very strong parallelism between this human-induced rapid artificial selection in the lab and what's going on in the wild. And this set of 1,500 SNPs is these SNPs are distributed all across the genome, and it looks like I'm about to run out of time. So I'll let you skip right to. I've been talking way too slowly, so I'll skip right to the conclusion, because what's the real kicker here is that there are all these parallel changes, but actually when we look just at each population separately, we see that it was a completely different part of the genome that changed the most. So basically we're seeing, even though there was some degree of a common parallel response, there were actually these really different, quickly. So basically here we're looking at the SNPs distributed out along the genome. We've mapped it to a closely related species. And we can see here along the y-axis is how the probability of an allele frequency change between generations 0 and 5. And here we see that in the up-selected lines, the major patterns occurred in these 2 chromosomes for one of the replicates, and nothing happened in the other one. And for the down-selected lines, there was an even more distinct contrast. So looks like I got to wrap up here. So basically, that's the conclusion. So what looked to be really parallel at the phenotypic level had a completely contrasting genomic basis. So that means that just studying the phenotypic aspects of fisheries-induced evolution might give us the impression that we have all these similar changes going on in different fish stocks, but it could actually have very, very different genomic consequences and very different degrees of allele frequency changes, and therefore loss of variation at particular genomic regions. So thanks very much.